one of the things that seems like it's a fairly rock solid part of the literature that I hear over and over again is, when we talk about nutrients that are good for your microbiome or that promote a healthy and diverse microbiome, the one that comes up the most is fiber. And you mentioned that fiber, things like cellulose, we don't digest them, but as people often say, they're food for the microbiome.
Mhm.
What I want to unpack that a little bit more. What do these microbes do with the fiber, and what are they producing that then turns out to be good for us?
Sure. Um, so this is not, I will disclose that this is not my area of great expertise. We don't study this a ton, but of course, it's one of the bigger things in the microbiome literature is diet. So the fiber there's lots of different types of fiber.
Mm.
So first I'll say that not all fiber is created equally, and not all fiber is going to be metabolized by the microbes equally, and not all fiber is going to have a benefit. So there's been a couple studies that I've seen where the fiber inulin, which is one of the ones that a lot of people will take, actually is really bad for certain diseases. It's good for some, but bad for others, based off of, how the microbes metabolize it and how it influences your immune system. But I think, one of the best metabolites that's studied because of fiber production is short chain fatty acids.
Yeah.
I don't know if you've heard of these before, but everyone studies short chain fatty acids. And these just seem to be globally good for you in that they induce an anti-inflammatory response. They can also act as fuel for your intestinal epithelial cells. So your intestine is lined with these epithelium. That's kind of one of the major cell types that creates a barrier between the microbiome and the host. And these short chain fatty acids are solely made by the microbiome, and they can be fuel for those intestinal epithelial cells and keep that barrier tight.
Yeah. Well, that's interesting. So these short chain fatty acids, maybe we'll talk about chain length later, but shorter, sort of smaller fatty acids, they can be used directly as fuel. It sounds like, so if you've got microbes in the intestine, they can take that fiber, make these short chain fatty acids. It seems like it's a really good, efficient way to locally have energy for those cells. They probably don't need to spend as much of their time and energy making fuel if you can have these microbes producing it locally right there.
That's right. Yeah.
And so what would be the difference between just at a high level? So when we talk about a short chain fatty acid being used for energy as opposed to something like glucose, is there anything worth mentioning there? Is like one of them better or preferred by our cells as a fuel source?
Glucose always seems to be the preferred fuel source for everybody, our cells and the microbes. So, I don't think that I can intelligently comment on whether or not short chain fatty acids or glucose are preferred. I think it all involves the environment, what the cell is doing, what the cell needs.
And I guess one last question I have here is, so they produce some of these microbes produce these short chain fatty acids, which can be used by cells for energy and perhaps for other things. Are there other ways to get short chain fatty acids, or is this the only way?
So our body doesn't produce them. It's, the fermentation byproduct of fiber that produces them. The other way to get them is people do synthesize these, like chemical companies synthesize them. So for instance, in the lab, we can buy, because there's lots of different short chain fatty acids. There's butyrate, there's propionate, and you can purchase these and you can orally take them. And a lot of people will do that in their mouse studies to show that, this short chain fatty acid is sufficient to have this effect.
But other than that, other than these products, is this not really a component of natural sort of whole food diet?
Not that I'm aware of. No.
So I know that there's, the microbiome's been connected to, all sorts of stuff. And we'll talk about some of that. Obviously it's connected to, gut issues that we can talk about, inflammatory bowel disease and things like this. But it's also been connected to obesity. And, we're going to talk about one of your papers that you shared with me that's really, really interesting here. But before we get to that in particular, what's sort of known, what's out there in the literature right now in terms of the connection between the gut microbiome and obesity?
So, what is currently known is that I guess very on a very basic level,
Hmm.
is that the microbiome, when it is less diverse, if you will,
Hmm.
is associated with increased obesity. So what does diversity mean? We have these we have these kind of like arguments in the lab about this.
Hmm.
But I think just on a very superficial level, if you are missing certain microbes, and we are still uncovering what these specific microbes are and what they do,
Hmm.
you seem to be more prone to gaining weight. So your microbiome composition seems to be very important in how you gain or lose weight.
Hmm. And in principle, like what would be the ways we imagine that could happen? So off the top of my head, I could imagine, well, maybe certain microbes are making certain key nutrients or producing metabolites that have to do with energy expenditure in our bodies. Maybe they're regulating how much of the calories we're actually absorbing from the food that we put into our mouths. What's sort of the space of possibility there?
Yeah. So I think one of the first mechanisms ever discovered was really from Jeff Gordon's group. Um, and this was when they basically took a microbiome from a obese mouse and transplanted it into a lean mouse, and that
Hmm.
animal gained weight.
Hmm.
And what they showed is that that microbiome was really good at extracting energy.
Hmm.
So it was able to extract more energy from the food, and then it gave more energy to the host.
I see. Did those mice actually eat more, or was all of it accounted for just by absorbing more?
In the transplant, the animals did not eat more. It was
Hmm.
more about, them just having more nutrients and
Hmm.
and extracting more energy. The obese animal used for the microbiome transplant, it was a leptin deficient animal, so it
Ah,
just
okay.
constantly eats. So,
Constantly is.
there's no doubt there's a component of, diet in influencing the composition of your
Mhm.
microbiome.
But just that microbiome change was accounting for a lot here.
That's right.
Yeah,
hmm
interesting. So tell us a little bit about this paper that you shared with me. Can you set that up for us in terms of what kind of questions you guys were asking and what the basic setup is?
Sure. So this was one of the first studies I embarked on when I started my own lab. So during my postdoc, I had been studying a bacteria that we had discovered was able to prevent inflammatory bowel disease in mice. And the mechanism by which it was doing that was inducing a T regulatory population, which is an anti-inflammatory cell. And that's how it was able to suppress inflammatory bowel disease. And we identified a pathway within the cell um, that it was able to induce this Treg population. So it was basically inducing it by making a polysaccharide that bound to a toll-like receptor on a T cell. So and a toll-like receptor is one of these receptors in innate immune cells that's able to recognize many components of bacteria. So they're
Mhm.
somewhat promiscuous.
So these receptors are for they're for sort of recognizing what is and is not a pathogen or a human cell.
They are, but they're broad in scope. So they
Mm-hmm.
recognize pieces of bacterial cell walls or bacterial DNA,
Mm-hmm.
and they can do that whether it's a commensal or a pathogen. So they're not really great at differentiating between the two.
I see. I see. They can just sort of see like this is our cell, this is human cell or not.
Yeah, basically, yeah. So people are still trying to understand how these cells help distinguish between commensal and pathogen.
Mm-hmm.
But it made us really interested in that pathway with respect to because it seemed like this pathway was really important in inducing a healthy homeostatic intestine. So we made an animal. So I should preface this, like I did not start my lab thinking I was going to study obesity. I
Mm.
was I thought we were going to, find a pathway that was involved in IBD. But you just follow the science. So we made this animal where we got rid of a signaling molecule that's downstream of toll-like receptors. It's called MYD88, and basically this is a signaling molecule downstream of most of toll-like receptors.
Mm-hmm.
by getting rid of it, you're essentially disrupting the ability of whatever cell type. We were looking at a T cell at the time, but you're disrupting the ability of that T cell to signal through any of those toll-like receptors.
So does that mean basically the immune system can recognize when it meets a non-human cell through this toll-like receptor, but it can't act on that because you're sort of blocking the information flow?
That's right, exactly. And I should say that we only knocked it out in the T cell. So the rest of the cells in the mouse's body were able to make that recognition. So they were still active. So when we made that mouse, one of the major phenotypes of that mouse is that it had a significant reduction in the amount of IgA it produced in the gut.
And you said we produce normally a lot of it.
A ton of it, right? But this mouse, when we knocked out this pathway, was not making as much. And then if you let those animals grow up to old age, most people do not let mice sit around in their facilities beyond 8 weeks because it's very expensive to keep mice. For some reason, my graduate student decided she was going to not cull any mice and she was going to let them grow up to be like over a year old. And these animals developed this spontaneous obesity. They were like really fat. They were like, she always called them like little pancakes.
But late in life, did they develop this late in life?
Late in life, yes. It was not immediately, we didn't observe it in like an 8-week-old animal.
I see. So as the mice are growing up and going through adolescence and things and becoming young adults, they looked normal macroscopically to the eye.
That is correct. Yes. So they did have this IgA deficiency at that early stage, but it wasn't until later in life that they developed the obesity. If you did put the animals on high-fat diet, they developed the obesity quicker, so you could start to observe it. But yeah, it seemed to be a spontaneous phenotype, which is very rare to see in animals, actually. Normally, you have to push the obesity by giving them a lot of fat or changing their diet in some way. So this was a really striking phenotype to us because we had knocked out this immune pathway. We thought it was going to be involved in gut homeostasis, but these animals were getting fat. So it made us think about the microbiota. I'm a microbiota lab, so of course we thought about that. So we did transplant studies where we took the microbiota that formed in these obese mice and we transplanted them into germ-free wild-type mice that did not have this knockout. And those animals got fatter.